Hi, everyone.
Hi, everyone.
Hi there.
Okay. Well, we have a very busy schedule today, so I'm going to get started, and welcome everyone. This is the HiRO Knowledge Translation Rounds. I'm David Lee, one of the electrophysiologists over in Halifax. And for anyone that's joining us for the first time, the HiRO Knowledge Translation Rounds are kind of a forum for researchers to present the latest research in the inherited heart disease space. And we particularly focus on research generated by researchers in the HiRO network and from data enrolled in HiRO studies. But in the same way that we have international speakers at our annual symposium, for this edition, we're really excited to have presenters from outside the network. As always, we hope that the presentations will focus on how the research presented may change our practice. And I look forward to some stimulating debate to help unpick that from the presentations that we deliver. So if I could, I'll hand over to Simon to give us some housekeeping details now.
Hi, everyone. So Simon Hansom, I'm one of the EPs over in Ottawa. As David said, really exciting lineup this evening. We've got three excellent speakers, and we're grateful to them for their time and for all of you for coming along as well. So a few housekeeping notes. The session is 1 hour, so we're going to try our best to keep to time. And that means we've got around 10 to 15 minutes for each presentation, and we're going to have a 5-minute Q&A following each person's talk. We really want to encourage you to participate. It makes for a really engaging meeting if you do. So for all the questions you're going to ask us, either raise your hand and then David or I will field those, or you can type in your question into the chat and then we'll do our best to put all those questions to the speakers. Without further ado, I'm going to go straight on and introduce Dr. Martijn Bos. Dr. Bos, it's my great pleasure to introduce you. Dr. Bos is a research scientist and associate professor in pediatrics, and he studies the basic genetic, translational, and clinical aspects of sudden cardiac death. He's a member of the Winland Smith Rice Sudden Death Genomics Laboratory of Michael Ackerman. And in addition to all the other things he does, he oversees the translational and clinical research studies of patients that they see in their genetic heart rhythm clinic. His recent focus has been on the development of AI algorithms for the diagnosis and management of genetic heart diseases. So he's going to present some of his work and his recent paper looking at deep neural network analysis of the 12-lead ECG and its ability to distinguish patients with congenital long QT from patients with acquired QT prolongation. Dr. Bos, thank you so much. I'll hand it over to you.
Thank you very much, Dr. Lee, Dr. Hansen, and I'm glad that this finally worked out. Dr. Lee and I have been trying to do this for a couple of times, and I unfortunately had to bail last minute for family reasons, but I'm glad this worked out. So keeping in mind on the time, I will get started. And as mentioned, I want to talk about the development and validation of artificial intelligence 12-lead ECG-based algorithms for QT prolongation. I'm actually going to present two papers today. I'm going to do a little bit of background on our first paper that led to the paper that Dr. Hansen just mentioned. And as you all well know, QT prolongation is fairly common. QT prolongation on the 12-lead ECG and clinically can lead to syncope, seizure, and sudden death. Now, there's two ways we can see that in the clinic, either as congenital long QT syndrome, or the prevalence of 1 in 2,000, caused by mutation in the cardiac ion channels, or as we would call it, acquired QT prolongation due to QT prolonging medications, electrolyte abnormalities, and comorbidities. And to distinguish these two in the clinic and to recognize them is a great challenge. So this is what we based our work on for this. Especially if you look at the 99th percentile QTc values by age and sex. And we can see that the current standards are over 480 milliseconds in women, over 470 milliseconds in men, and we guide ourselves by under 400 or over 460 milliseconds in boys and girls under 13. And as you can see in these diagrams, there's a significant overlap between the normal QT values in that 99th percentile and long QT syndrome. Furthermore, what we have seen is that in our clinic, in a large study we presented about a decade ago, that about 10% of our patients presenting to Mayo Clinic had isolated QT prolongation over the 99th percentile threshold. Now, obviously, those are not all long QT syndrome. That would make it very prevalent, even though we are a referral center for long QT. So there's other reasons why they're there. These patients are probably sicker, have a higher frequency of electrolyte and other abnormalities, but makes it very important how to distinguish these things. Which made us wonder is, can we use AI for this? In our first study, we wanted to determine whether an artificial intelligence deep neural network can aid in the diagnosis of long QT and distinguish them from patients with acquired QT prolongation. Now, as you all well know right now, AI is all around us, and I want to do a little bit of a practice exercise as I prepared for this. And, we see in Google has AI overviews when you Google something now. It is in Microsoft, it's everywhere. And, in the last few years or before, I wanted to see if I could have AI generate me an image for this talk and create me a heart and an ECG strip. And before AI, I had to go to my media department, go back and forth, and take a couple of months, pay them internally. But we have AI. But for AI, you have to be very clever and very creative. So I could say I want a artistic publication-ready anatomical drawing of a heart with an ECG strip showing prolonged QT interval. But to better the image and to get the best image you want, it should be much more comprehensive and creative, and I'm not that. So I use this AI prompt generator, and it shows me, and I won't repeat all of this here, but it asked me to create an intricately detailed showcase of the chambers, valves, and major blood vessels, and an ECG strip displaying a prolonged QT interval and accurate waveform. And within about a minute, when I went to my friend ChatGPT, which has a image generator built in called DALL-E, it gave me an image, and I was pretty impressed. But right away, as you might notice, and all of you should, AI is not perfect, because I did think these valves looked a little funky. We were gifted another R wave, and our intervals, especially our QT interval, became really, really long. So yes, AI is everywhere, but there's some pitfalls that we have to keep in mind when we do this. So for our first study, we asked ourselves, can AI ECG aid in the diagnosis of concealed long QT syndrome? This paper was published a couple of years ago in JAMA Cardiology, and I have the reference for you here if you want to look that up and read it again. But what we wanted to do, and I have to give a disclosure here, we is some really smart analyst and scientist in AI, and not myself, is build a neural network to see if we can, A, distinguish normal patients from patients with long QT syndrome, and whether we could distinguish the three major subtypes, type 1 long QT syndrome, type 2, and type 3, caused by the potassium channels for 1 and 2, or the sodium channel in 3. So for that study, we took all our patients seen in genetic heart rhythm clinic, as about 3,000 at that time, and we found all the patients either with a diagnosis of long QT syndrome or those who were dismissed normal. And these will serve as our controls, and we call them dismissed normal, or sometimes we've called them hypercontrols, because these patients might have a little bit of an abnormal ECG. There was a reason why they were evaluated. They were left our clinic with not with the diagnosis of long QT syndrome and pretty much normal, but there might be something there. So there's a disclosure of the normalcy of these patients, but they were served as our controls for this study. And we built two models. One was the long QT syndrome detection model, and two, a long QT syndrome genotype model. And I'll be very brief on this model. Like I said, we had very smart analysts and data scientists build these models, doing things that I cannot replicate for you. But basically, the idea is, can we take this 12-lead ECG and we change it into all numbers in a matrix of 12, the 12 leads, by 5,000 data points? Now, for me, as a simpleton in the AI, I would say that is 60,000 data points. The AI model will correlate these 60,000 data points to itself. That's 60 to the power of 60,000. And then compare this for the group. So the power of the AI is what you and I can see in this AEG magnified by multiple factors. And the way to then read out whether this model can or cannot do that is the receiver operator characteristics. And what you see here on the left is that indeed the neural network in the blue line was significantly better than the QTC alone in distinguishing long QT syndrome from normals. And an AUC of 0.9 or 90% is a very strong model in that. Additionally, it also was able to distinguish the three genetic subtypes from each other with very strong AUCs over the 0.9 threshold. Now, obviously, when you look at this, you could say, well, yeah, the model can do this, but I can do that too. On the left side, patients with long QT syndrome should have a prolonged QT. And on the right side, we know that each of these long QT syndromes have their characteristics in their ECG and in their T waves, especially, that distinguish them from each other on the genotype. So our next question was then, can we distinguish patients with a concealed long QT syndrome from those that are called normal? And we know, as you see, a little bit of a small print there, is that a large number of patients, about 30 to 40% of our patients, have a QTC that would not be diagnostically called long QT syndrome, but their genetic predisposition, their phenotype, their features, their family history have shown that they actually have that diagnosis. And then looking at that and comparing those with a normal QTC called normals under 450 milliseconds, or those with long QT syndrome and a QTC over 450 milliseconds, we could see that the neural network, again, was significantly able to distinguish patients with a concealed long QT, even when the QTC is normal. So model one, in our opinion, fairly successful. If you come to a clinic with a suspicion of long QT syndrome, the ECG or a neural network, even when your QTC is normal, can help you in diagnosing long QT syndrome. But then again, as I showed earlier, there's a large number of patients that come to our clinic and to many clinics with QT prolongation for other reasons of those that we call acquired QT prolongation. So in this, our objective was to determine whether a similar model can distinguish patients with long QT from those with acquired QT prolongation. And this is the paper that Dr. Hansom mentioned. This is a paper we published last year in Mayo Clinic Proceedings with the reference right here. And we used a similar setup. We have now built a cohort of over 1,600 patients with long QT syndrome with at least one ECG done at Mayo Clinic. So we had the digital file available. And then for our controls at this point, we used the Mayo Clinic ECG data vault of over 2.5 million patients with at least one ECG. We used the guideline thresholds for QT prolongation and found all patients with a QTC over the thresholds listed here. And that in our cohort identified for our controls in the data vault over 360,000 patients with a QTC over the threshold for a total of almost about 1 million ECGs, and I've shown about 14%, so over that 10% that we had shown in our earlier study. We then built two cohorts, a long QT cohort, 808 patients with long QT, showing the concealed patient with concealed long QT had fallen out, and 3,500 controls patients. And what we did is we made a match where we matched the patients versus controls at a 1:5 and a 1:10 ratio. And then, because we know that patients with a pacemaker or a wide QRS could bias the model, could bias the QT measurements, we did the same analysis on patients with long QT in a smaller cohort and controls, again, at a 1:5 and a 1:10 match. Just for frame of reference, for those who want to study this, here are the model development parameters. I won't go into detail because of previous disclosures. But when we looked at that at a 1:5 match ratio, we show that this ECG model, AI ECG model, was excellent in distinguishing patients with long QT versus those with acquired QT prolongation. And as you can see in blue, were those all the patients, and then in orange the patients after excluding those with the pacemaker or bundle branch block. And at a 1:10 match ratio, that was the same. Again, we tested these ratios to see whether one would be better than the other, but we did not see a significant difference or no difference at all in the receiver operator area under the curve. But again, these patients, this is a arbitrary match ratio, if you will, at a prevalence of 1 in 2,000 for disease, or a 10% presentation of long QT at a clinic. That's not really yet a screening model. So what we wanted to do is test the performance of the model at clinical screening levels, considering the disease prevalence of 1 in 2,000. So for that, we restructured the cohort, and if you look at the bottom, we did two things. We selected our controls now to have either a 1 to 200 ratio or 1 to 2,000 matched model development. And the reasons we chose those two numbers were twofold, or were two reasons. For the 1 in 2,000 is the prevalence of the disease. For one patient, there would be 2,000 controls that should not have it. But as I previously said, there's 1 in 10 patients present with long QT syndrome. So we picked a 1 in 200 to reflect the clinical presentation of QT prolongation in this cohort. Did the same development of the model, same comparisons, and again, excluded patients with a pacemaker or a wide QRS. And when we did that, on the left side here, 1 to 2,000 match ratio of long QT prevalence, trying to mimic a clinical or a real-life scenario, we see again at a AUC of 0.933 that the model was excellent in distinguishing patients with long QT from those with acquired long QT or QT prolongation. And similarly at the 1 to 200 match ratio. Again, this is not necessarily a screening model, but at least an attempt in a model creation to create those settings. So in conclusion for this, AI ECG models can successfully distinguish long QT syndrome from healthy controls, even when the QT is normal or concealed, and from acquired QT prolongation. But we have to recognize that AI ECG models are not perfect. They need to be further trained to serve as true screening platforms and validated in outside cohorts. But, and like the weaknesses that we saw for the image generation, and when we recognize that they are excellent tools to aid in diagnosis and management of cardiovascular diseases. Thank you very much, and I look forward to any questions.
Thank you. That was really a wonderful presentation and some really exciting research there. To kick off, I'm curious to know a little bit more about the actual genetic distribution of the population that you studied. So was this just drawn from the 3 main genotypes, or did you have a more diverse population of sort of rarer genes involved? And yeah, as a first question.
Yeah, great question. So the 3 main subtypes make up about 75 to 80% of genetic long QT syndrome. So for these models, we focus solely on the long QT 1, 2, and 3. The other long QT 4 to 17 or 18, whatever number you want to call long QT, are so rare, even in our clinic, and have so much different biological and pathological background that we left them out for now. Because in the end, as we saw in the long QT model in our first paper, there is similar, or there are different signals. So the only thing we would have been able to do is, and we have done that with some of our clinical papers, is lump them all and say these are the minor genes. But recognizing the underlying pathological or the physiological backgrounds, we decided not to do that for these.
Mm-hmm. And then sort of following on from that, obviously that there will have been a few ECGs that were, I guess you might call them false negatives. Was there any particular trend in a particular genotype that was more prevalently misassigned?
Yeah, so we haven't went back and looked at what are true positive, false positive, what the genes were. So right now it was mostly the model development, but we haven't looked at those trends as you suggest, but it's a great question.
And I guess finally, unless Simon, do you have anything from the chat?
Nothing on the chat yet, so encouraging people to please type in or raise a hand to ask a question. That would be fantastic. I mean, I might ask a question if that's okay. I think one of the things that really interests me is that you can have a shared mechanism of action. So, we know full well that some drugs can interact with IKR and mimic long QT2.
Yep.
So how the heck does the AI tell the difference between the same mechanism?
Yeah, so we started some modeling for our long QT paper is what's called SHAP modeling, so that we can see which area of the ECG is there. We see some QRS differences and some PR differences that they are. We haven't done that for the acquired yet, but you're totally right. The potassium channel effects of some of these drugs are a key factor. And I think our next study would actually really expand the QT prolongation cohort, draw from the electronic medical record their drug status, their electrolyte status, and see if we can group them and separate them, because right now they are lumped, right? It is QT prolongation for any reason that is not long QT syndrome. And to really say, okay, these are drug-induced. These are, and within the drug-induced, can we separate them between those that we are known are HERG blocking or HERG affecting drugs versus the non? So that will be our follow-up study that we're working on as we speak.
That's great. Thank you.
Right.
And in follow-up, that would be the ideal, right? Is to help the non-EP, but even the EP physicians to say, here's your ECG as a tool, and give you the answer. There is QT prolongation. Well, we should be able to pick that up, but here's the possible reason why, and then guide towards something that is treatable, or we can withdraw a drug or treat the electrolyte imbalances, or saying there might be a hint that this is congenital, go this path. So really advise on that first ECG.
Fantastic.
Thank you.
Thank you so much.
So with that, it's time to move on, and I'd like to introduce Dr. Alessio Gasperetti. So Dr. Gasperetti was born and raised in Milan. He graduated from the university there in 2018. Prior to his intern year, he completed a postdoctoral fellowship at the University of Zurich, and subsequently spent 3 years doing an ARVC fellowship at Johns Hopkins under the supervision of Dr. Calkins. And his research interests lie in electrophysiology and cardiomyopathies, specifically in arrhythmogenic cardiomyopathy. And he'll be speaking to us today on relating to his work on DSP-related cardiomyopathy. So,
thank you for having me. Really appreciate that. Let me see if I can actually share my PowerPoint presentation properly. Seems okay. Is that okay? Perfect. So thank you so much. As mentioned, I'll be talking briefly about DSP cardiomyopathy, and those are my disclosures. But for me, DSP cardiomyopathy is a very interesting and unique form of cardiomyopathy. This is a family that I saw when I was a medical student during my last year of medicine in Italy. And the part that really struck me looking at this, that ended up being a family harboring a truncating variant in the DSP gene, is that within the same family, we are having so many different phenotypical forms, with patients presenting with DCM, patients presenting with classical ARVC, patients presenting with ALVC, and red arrow is our proband, but also many, many patients that ended up having episode of myocarditis. So within the same gene, we had really a huge amount of different clinical presentation, and I found that fascinating. DSP cardiomyopathy is sort of a novel entity. It has been described as a unique and independent entity starting in 2019. This is a multi-center paper coming out of an effort led by our colleagues in Michigan, but several very high-end cardiomyopathy centers participated in here. And this disease was sort of described as mentioned, with a disease with significant phenotypical variability, with sort of a pathological hallmark which was the presence of inflammation and LV scarring. It was a pretty nasty disease with a very high morbidity in terms of VA and heart failure rates, a disease for which was very difficult to perform some risk stratification, and as mentioned, a disease that's strongly associated with myocarditis. This is sort of where I started working with DSP. The points in red are the points that I will briefly touch. And in 2020, to sort of answer some of those questions, just because there was not that many data available, we started something called the Desperate Network, which was a gene-first effort to characterize the clinical characteristics of DSP cardiomyopathy. This was coordinated by Hopkins, but we ended up enrolling as many patients from as many centers across the globe as possible. And it ended up sort of skyrocketing. We started with 28 centers and around 1,000 patients, but at the last check, we ended up having 43 centers with around 1,500 patients across the globe. And so I will quickly move through some of the findings coming out of the Desperate Network. Is a series of paper at this point, just going through what's mostly clinical relevant for EPs and cardiomyopathy specialists alike. The first point I really want to touch is the phenotypical variability of this disease. This is a sort of distribution of phenotypes coming out of our first paper, which was a paper published in European Heart Journal 2025. 800 carriers of DSP variants were enrolled in this study. And as we can see, there is sort of a pretty even split between a third of those patients presenting with classical ARVC right-sided task force 2010 positive phenotypes. A third of those patients will present with either a DCM or a non-dilated left ventricular cardiomyopathy phenotype coming out of the most recent ESC guidelines. And a third of those patients will present with no recognized phenotype. Regardless of the phenotype, all of those patients end up having pretty high rates of ventricular arrhythmias and heart failure. Clearly, patients with DCM or non-dilated left ventricular cardiomyopathy end up having more heart failure propension, while patients presenting with ARVC phenotypes end up having a little bit more of arrhythmias. But all in all, a pretty aggressive disease. And the part that I think it's always worth remembering is that even those patients that present with no phenotype at all end up having 0.5% chance of having a sustained ventricular arrhythmia event per year and 0.3% of heart failure hospitalization per year in the absence of phenotype. So really pointing towards not just phenotypical variability, but a significant penetrance of disease in the presence of a variant. Another pretty important point is that for those patients, there was a long conversation about what's the best way of performing arrhythmic risk stratification. For most patients with the RVC and ACM, the most used tool these days is the RVC risk calculator, published in 2019. That, however, it's a risk stratification tool that works really well for classical right-sided disease and does not seem to work well for left-sided or biventricular forms of this disease. When this was tested by multiple cohorts, the performance of this tool was tested in patients with DSP on the left in a small cohort by our colleagues in London, and on the right on a sort of genotype mixed phenotypical cohort having biventricular and left dominant forts of ACM. And as you can see, the predicted risk, which is generally the curve on the top, on the left it's the red curve, on the right is the curves with the triangles, is always much above the observed curve, really showing that this risk stratification algorithm does not work well. What we did using the Desperate cohort, we ended up enrolling around 250 patients that fully qualified for the use of the ARVC risk calculator, so task force positive with no prior ventricular arrhythmias event at the time of first inclusion in the study. And we really tested how this performed over 5 years. And as you can clearly see, regardless of the presence of LV involvement, on the left, the performance in patients with the LV involvement, on the right, the performance in patient without LV involvement, so the most classical RV dominant forms with DSP variants. And in either of those settings, the risk calculator ended up not working particularly well, with a statistics that on the left was in the ballpark 0.5 to 0.6, and on the right, it was 0.62. So really not working particularly well for those patients. This led to our next project, which was sort of a dedicated DSP-specific risk stratification algorithm. This was a manuscript that was published at the end of last year, where we ended up using 451 patients to develop using a multi-step regression, Cox regression. We ended up building a specific risk calculator to predict the 5-year risk of a patient to develop sustained ventricular arrhythmias. The risk factors that were identified were sort of unique. Specifically, on the left, those 5 risk factors are listed, namely female sex, presence of non-sustained ventricular tachycardia, the PVC burden of those patients seen at a 24-hour Holter, the presence of a LV dysfunction, defined as an ejection fraction lower than 50%, and the presence of a moderate to severe RV dysfunction. The part that is very interesting is that some of those risk factors are shared by DSP with all the other forms of arrhythmogenic cardiomyopathy, but some are unique. And specifically, the female sex is something that we have seen as a very important risk factor in patients with DSP, but it's also sort of a unicum within the HCM, the arrhythmogenic cardiomyopathy phenotype, as this is the only gene described so far having a predominant increase in risk in female patients, where in most other genes, PKP2 being the most clear, or LMNA or TMM, male patients are at much higher risk. This is currently under the effort for external validation for this is currently undergoing, and we hope to have a presentation at HRS this year with a published manuscript. But just to give all the colleagues an idea, this ended up performing much better than the other 2 that we have, with a C statistics in the ballpark of 0.75, which is what we were aiming with this sort of risk calculators. One final unique characteristic of these patients that I think it's worth mentioning is sort of the presence of those episodes of myocardial injury, of myocarditis. So flare of chest pain associated with troponin leakages that have been associated with more active phase of disease. The reason why we really end up hammering on this topic using the Desperados network is that there were multiple reports showing that the risk profile of patients with DSP tends to be significantly different before and after the occurrence of those myocardial injury episodes. As you can see on the right, those are time-dependent Kaplan-Meier curves. So one patient can be at the same time on either curve, depending on the natural history of the disease. The patient is on the blue line before experiencing any myocardial injury episode, and instead their risk profile moves to the red line after experiencing a myocarditis or myocardial injury episode. As you can see, there is a significant difference in risk profile with an increased amount of ventricular arrhythmias and increased amount of heart failure hospitalization after the occurrence of those episodes. And while the pathogenesis of this is not fully clear, there is with the highest likelihood, this is probably driven by inflammation leading to increase in scar and worsening, increasing the activity of both triggered activity for arrhythmias or inflammation leading to more heart failure hospitalization. And then coming to our most recent paper, specifically addressing myocarditis, we ended up not just looking at what the overall prognostic role of those episodes was, but also if there was anything actionable regarding those specific episodes. And there are probably 2 main take-home messages from these papers. We ended up having around 1,400 patients in total, with around 250 patients experiencing a myocarditis at some point. The first take-home message that I thought it was really important was that there is sort of a dose dependence relationship between the amount of myocarditis episodes and the overall risk, with the idea that the more myocarditis episodes patients experience, the higher the overall risk of VA or heart failure. And as you can see in the curve on the right, the risk tend to be higher and higher at the occurrence of repeat myocarditis episodes. But the other important part very important hypothesis that we tested in this manuscript, and again, with the main caveat of these data being retrospective and mostly hypothesis generating, was that we wanted to see if patients that were treated with immunosuppressant medication around the time of myocarditis events had the same overall arrhythmic risk and heart failure risk of those patients that instead had the myocarditis episode and were not treated. So in those 2 curves, I recognize that this slide is a little full of information, but the red line represents the clinical risk of arrhythmias on the left and of heart failure on the right of those patients that had a myocarditis episodes that was treated with immunosuppression. In blue, those patients, this is the risk profile of those patients that had a myocarditis episode and received no treatment at all. In yellow is the risk profile of patients that had a myocarditis episode and were treated with NSAIDs or colchicine. And in black, this is the risk profile of what we define as controls. So Desmoplakin carriers that had absolutely no myocarditis episodes ever. The take-home message from those two images is that the risk profile of those patients experiencing myocarditis episodes that undergo immunosuppression seem to be in line with DSP patients that never experience myocarditis episodes. So again, recognize that this is retrospective and hypothesis generating. The idea here is that what we're seeing is that the risk of the additional risk of VA and heart failure that comes with those myocarditis episodes is sort of negated and blocked by the use of immunosuppressant. We've clearly recognized that we need a randomized control trial to fully validate and support this data. Coming to the final point that sort of really merges into the specifics of DSP cardiomyopathy is the response of those patients of exercise. This is really important, as we all know that arrhythmogenic cardiomyopathy and the right forms of arrhythmogenic cardiomyopathy tend to be really worsened by endurance exercise in general, with exercise acting as a risk factor for ventricular arrhythmias, heart failure, hospitalization, and need for transplant. When we tested the role of exercise in DSP cardiomyopathy using 100 patients carrying DSP variant, for which we had extensive exercise data from birth to our final assessment, we really showed that there was no direct interaction between exercise and arrhythmias, but it was a pretty significant interaction between exercise exposure and the occurrence of myocarditis, really pointing towards the modulation of inflammation in these patients more than a direct effect. I recognize I really went through most of DSP research of the past years, so I'm more than happy to take any question.
Alessio, that's a fantastic overview, and well done for all the hard work in the field. I share your fascination with DSP. I think it is fascinating. Do you think, or have you seen that there's any correlation between the phenotypic presentation, so right side versus left versus DCM, in patients who then present with a myocardial injury pattern? So is it more common in people with left-sided prevalent disease than it is right, or?
I think that we are sort of in the circular argument here, because the main problem that we have is that the phenotypical descriptors that we have are sort of, most patients that come with myocarditis will have some degree of late gadolinium in the left side. And according to the new ESC guidelines, those count as non-dilated left ventricular cardiomyopathy. So clearly there is a strong association between the two. Yeah. Just because someone comes in with a RV phenotype, it tends to be not super common. I wouldn't discard the presence of those myocarditis episodes. Probably overall a little bit more common in LV dominant forms. But this just may be the way we classify those patients rather than through biological association.
And interesting, you showed a beautiful figure of the benefit of immunosuppression in both groups for VA suppression and freedom from heart failure. Interesting that colchicine and non-steroidal seem to maybe have less benefit in VA reduction, but seem to have maybe some benefit in heart failure.
I just want to say the numbers are relatively small. So it ended up being non-significant for either. And that was one of the big questions. Honestly, one of the questions that we had as co-authors when we were writing the paper is, how do we even classify colchicine? Because some of our colleagues have historically classified it as immunomodulator, maybe not suppressant. But regardless of the way we did, we had a multi-state model. We ended up correcting for all possible correction that you can think of. Really, colchicine and NSAIDs end up having a null effect on those patients.
Beautiful, thank you. Tom Austin, I can see, has his hand up. Thomas, do you want to ask you a question?
Yeah, that'd be great. Thanks, Alessio. That was a wonderful presentation. And obviously, your work in this area, I think, has been critically important. I'm just wondering your take on two things. The first is, what is your threshold to prescribe immunosuppression? So how convincing does the myocardial inflammatory episode have to be, and how many do they have to have before you consider it? And then my second question is, if you are going to start immunomodulators, do you have a first-line approach to this, like whether you start steroids at the same time, which immunosuppressive agent you use, and who follows these patients? Is it rheumatology or is cardiology doing all its own? So I know those are several questions rolled into just a couple, but I'd be grateful to hear your thoughts.
And I think, it's a spot-on question, that it represents one of the critical problems that we're currently facing. I'll start from the end. I am lucky. I'm currently working at Hopkins, and I have a heart failure department that really is really intertwined with our arrhythmia clinic. So most of the patients with DSP that are seen in arrhythmia clinic will end up seeing heart failure maybe the same day, and they are very comfortable using immunosuppression in general. Now, what patient do I recommend immunosuppression? And again, we are in a sort of a data or maybe not data-free area. I can give you my personal take on this. We're outside of any guidelines right now, so it's really case by case. I think that repeat myocarditis episodes, so the second myocarditis episode in someone that I know having DSP, it's when I start considering. Most of the data that we have comes out of a steroid-less regimen. 80% of the immunosuppressed patient in the paper in circulation and were on a prednisone-based regimen that ended up being either prednisone alone. Most colleagues end up doing 1 mg per kg and then ended up tapering over 3 to 6 months, or they ended up using the TIMIC protocol. Most of our European colleagues so ended up doing prednisone plus azathioprine. There is an American enclave that has been using prednisone and MMF quite extensively. One of the main problem here is that mycophenolate it's teratogenic, and as with most of those patients are fertile female, sometimes it comes with pretty heavy problems in terms of contraception. I think that one final line that I always like to mention, for which I have no data in terms of changing long-term outcomes, but we have been using more and more in recent times, especially for patients who have chronic chest pain and chronic troponin leakages, is ILR modulators. So in the United States, we have anakinra and rilonacept. I know that rilonacept is not available in Europe. I'm not sure if it's available or not in Canada. But what I've seen is that those work really well for symptomatic management. We had, I think at this point, 14 or 15 patients treated with rilonacept that went from chest pain constantly every day to be basically chest pain free for this point 6 months. We need a trial. That's the correct answer.
That's a great answer.
And can I ask a follow-up question to that? It's obviously very helpful to hear what other people are doing. Do you see any role for PET scanning, either before or after or during immunosuppression? And then just thinking about the prednisone taper around that, because obviously these are young people that you don't want to leave on high-dose prednisone for more than a few months if you can avoid it. So is there a path that you have where patients, for example, use PET scanning to come off of prednisone and transition into a steroid-sparing agent, or you just do it sort of ad hoc based on what you feel is best?
Unfortunately, the second one, and I'm saying unfortunately because I think that moving forward within the Gasperetti's network, we really need to try to standardize the approach, because that's the best way to get the cleanest data. It's very difficult for me at Hopkins to get PET just because our nuclear medicine doesn't have a ton of availability. One of the main caveats is that there are a few reports from specifically in arrhythmogenic cardiomyopathy out of Zurich showing PET positivity around the time of ventricular arrhythmia events and or increased PVC burden in those patients. So I'm always a little skeptical of PET in those patients, because I think that the rates of false positive, or at least rates of positive with tracing capturing in times of potentially not real inflammatory activity, but more arrhythmic activity, can be high. I don't have the data fully sorted. So right now we're not using it, but I think it's not unreasonable. Especially, I think it will be super helpful in moving towards a steroid-sparing agent. We try not to keep anyone on a dose of steroids, an elevated dose of steroids, more than 3 months. So we're really trying to taper, acknowledging that those are relapsing, remitting episodes. Immunosuppression do not prevent future myocarditis episodes. What we are doing is risk mitigation of the consequence of the inflammation. We're not preventing further inflammation just because that inflammation is genetically determined.
Yeah, that's absolutely fantastic. Thank you. Thank you for your insights and expertise in the field. Just in the interest of time, so that Christian has enough time for his presentation, finally, I'm going to introduce Christian Steinberg, who's well known to all of us. So he's an electrophysiologist and the lead for the Inherited Arrhythmia Clinic in Laval, Quebec. He's a long-term HiRO member, a friend and colleague, and his research is focusing on inherited arrhythmia, applied cardiogenetics, and complex arrhythmia. He's going to present his work on TREC1 targeting autoantibodies in patients with short-coupled VF. Christian, it's great to have you, and over to you.
Thank you very much. First of all, I have to apologize. I have a very bad bronchitis, and my voices are almost completely gone. So please apologize upfront for that. I do my very best. So I had already the privilege and the occasion to present on the first part when a few years ago we could, in the combined HiRO pan-Canadian effort, establish short coupled ventricular fibrillation as a distinct electrical disease, distinct phenotype. And the second part is what we still do not know is what it actually causes it. And one of the most recent hypotheses might be, could it be an autoimmune channelopathy? As you all know, like SCVF, as we abbreviated, has hived off of this large pool of IVF, which was initially the mother presentation form of all these many primary electrical diseases like Brugada syndrome, CPVT, and short QT, which we could all distinguish. And the most recent one of these primary electrical disorders is SCVF. And what it is it's difficult to identify because there's no distinct ECG signature. It's a primary electrical disorder. The resting ECG will always remain normal. You can nothing provoke by an exercise treadmill test or even when using a burst protocol or whatever. And the hallmark is the VF onset by a trigger PVC with a very short coupling interval that is usually less than 350 milliseconds. And in the true case, it's actually around less than 300 milliseconds. And the first clinical manifestation is typically cardiac arrest or sudden cardiac death. And we could initially show, and most of you are familiar with this, and you've participated in this study, that SCVF is a distinct phenotype, and it is actually increasingly frequently diagnosed since we know a little bit what we have to look for. So at least 14% of unexplained cardiac VF arrests are related to SCVF, and it is estimated that the case-related fatality is probably at least 25% because obviously there's a survival bias. So and when we established this using our Canadian HiRO/Casper data, we also desperately did in-depth research for a genetic cause, but we couldn't find anyone. So so far there's no evidence of a robust Mendelian genetic substrate. And there is a specific subtype in the Netherlands that is related where you can find DPP6 risk haplotype and the non-coding part. But even in this Dutch IVF cohort, only 48% of these patients actually have then a correlation between DPP6 risk phenotype and SCVF. We also all know that quinidine is the only efficient antiarrhythmic medication, and there's some reports that verapamil as alternative or add-on has some efficiency. And we are also all familiar, this is a very aggressive disorder. Actually, when I'm asked, so which kind of genetic cardiomyopathy or primary electrical disorder scares me, I usually answer it's SCVF. This is clearly the killer. Forget about CPVT or everything else. The highest reported VF recurrent rates is SCVF. 79 to 92% of patients will at least have a second VF episode within 5 years after the index event. And at least 1 out of 5 will present with an electrical storm at the index cardiac event. Definitely no other cardiomyopathy or electrical disease comes even close to this. And obviously, to document it, you have to exclude everything else, and you need the VF onset. This is one of the reasons why most patients, roughly 3/4, 72%, are only diagnosed at the time of VF recurrence. And SCVF also has some striking overlap features that is sharing with other primary electrical diseases, including Brugada syndrome, short QT, or Ripoll, what is common to all these four disorders is that the VF is initiated by trigger PVCs with a short coupling interval. The VF typically will manifest at rest or during sleep, and they respond to quinidine. However, in contrast to the three other diseases, SCVF has no distinct resting ECG phenotype. And then what we tried is we did whole exome sequencing during our Canadian cohort, and we couldn't find anything. And we were very disappointed. And the question was, so is it something polygenetic, or is it something that is not genetic at all? And so we actually looked outside of the box and said, well, maybe we have to look for non-genetic mechanisms. And this is what we did. In this, I will call it like a pilot study that was just our center here in Quebec City. We took a small cohort of true SCVF, 14 patients, 19 patients with IVF, and we had 38 matched healthy controls. And we said, well, maybe there's something else. And then we were looking, the next obvious big field to explore is autoimmunity. So we took plasma samples and we were screening to see if there's any autoantibody signature. And in the case, if we would do something, well, then we would perform function experiments. So what we did is we created a library of all epitopes of all cardiac ion channels that are in the cell membranes of our cardiomyocytes. You can here see the different epitope sequences on the right side. And then this test functions a little bit like an ELISA. You have coded wells that will be incubated with a diluted patient plasma, and then you direct an human antibody with a fluorochrome, and you see what you get. And what we found is that 50% of the SCVF patients that had really true diagnostic criteria of SCVF had a circulating antibody directed against the channel that we will discuss in a second that is called TRAC1. We also find this 10% in the undifferentiated IVF and 10% in the controls. The controls were unrelated. There were no relatives, so it's a little bit difficult to explain, but this still remained significant. So then the question is, what is TRAC1? And TRAC1 is a little bit like the lost child of cardiac electrophysiology. It is part of a very special family of cardiac ion potassium channels called so-called 2-pore K2P potassium channels. It has already been detected in the very 90s, and it is in the cell membrane, but it is a minor player. So on the left side, you see the usual suspect that we know from our different inherited arrhythmia syndromes, the voltage-gated potassium channels that have a typical 6 transmembrane segments and the inward rectifying potassium channels that have only 2. But the common thing is these 2 families of potassium channels have only 1 pore. The so-called 2-pore potassium channels have 2 pores, so 2 channel lumens for potassium ions. It's a whole family. And TRAC1 is also abbreviated as K2P 2.1. It's a very long, complicated name, and it is expressed in the human ventricle. And it does also in different rodents and other mammals express it. It is in the sinus and AV node and also in the atrial and ventricular myocardium. And for many years, it was a little bit out of focus, but over the last few years, there have been a few isolated reports. For example, there has been a case series of a family with RVOT-VT related to a distinct missense mutation in TRAC1. It is also known that in heart failure, it doesn't matter of what origin. There is, as part of the electromechanical remodeling, a downregulation of TRAC1, and this explains in part shifts of the resting membrane potentials. And so the antibody that we found is, it is an IgG antibody that is directed against the SCAP part of the channel. So it doesn't react with the other epitopes that we tested. So you have the 4 in purple, the 4 transmembrane segments, and the pore helices P1 and P2. And in green, there's a little extracellular cap structure that is important for the dimerization of the mature channel, and it also directs a little bit like a rudder the potassium ions towards the lumen of the two pores. So the mature channel is actually a homodimer of two of these structures, and the anti-TRK antibody is directed against the cap form. So then it's always important, we all have hundreds of and dozens and hundreds of circulating autoantibodies, and most of them don't mean anything. So is it functional? So we express the channels first in a HEC293 cell model to see if this antibody has functional properties. And yes, it increases the properties of the channel. It causes a gain of function effect. It significantly increases the potassium flow through the channel that you can see here in the current voltage dependent here in green, and in dose dependent. If you add the antibody, the channel is significantly activated compared to the baseline here in gray. And if you add quinidine, then you antagonize the effect of the autoantibody. And the next step, then we said, okay, let's test it in iPSC-derived ventricular cardiomyocytes that we took from healthy donors. And what we found is that you can see here in the top figure of B is MDP, is the resting membrane potential that is unaffected from this antibody. So in gray is the controls, and then we have different concentrations of the antibody. But concentration dependent, you increase the amplitude of the action potential, and even more important, you significantly shorten the refractory period. And this extremely shortening of the refractory period is actually the hallmark of the VF onset in SCVF. And when you antagonize this with quinidine, then you can restore to a great extent the duration of the original action potential duration. What you can see here, like in blue, the quinidine bars can reverse part of it. And what we can also see is that in these myocytes, upon the exposure of anti-TRK1, they create those spike and dome-like in green action potentials, which become neutralized by quinidine. And this spike and dome-like action potential has a striking resemblance with another disease that we all know. We know that TRK1 has a transmural expression gradient. It's more expressed in the endomyocardium and less in the epimyocardium. And when we did this first experience, then we said, damn, this looks exactly like the mirror image of another well-known suspect that has an transmural gradient from epi to endo. And this is, as you guys probably all guessed, is Brugada syndrome. In Brugada syndrome, for the ITO current, so KV 4.3 and 1.4, in the opposite around. So it just something like this. When we looked, then is the presence of these autoantibodies related to a specific disease severity. So in our sensitivity analysis, it was strongly limited by the small sample size, is although all our pairwise comparisons were non-significant, we nevertheless observed that those patients who had electrical storm or a very severe phenotype, so more than 2 VF recurrences within the first 2-3 years, had 71% had the TRK1 antibodies compared to only 14% who had only a single episode. And then the question was, right, what does cause these autoantibodies? And said, all right, TRK1 is encoded by the KCNK2 gene that's actually not covered by any panels in research or standard in heart arrhythmia. So it's a relatively large gene on chromosome 1, has 7 exons. And we did an entire gene sequencing, including these huge intronic sequences. This is over 154,000 base pair gene. And unfortunately, nothing. No pathogenic variant. That would be like the low-hanging fruit. Is a micro variant that causes some immune exposure of epitopes. So the open questions are, first of all, why do these patients have the anti-TRK1 antibodies? Is it just a specific subgroup? We just tested it in French Canadians, and we just observed it in 50% of patients. Is there in the other ones? There were SCVF2, but is there a time-dependent fluctuation or a seroreversion? Is it truly a marker of disease severity? And do our patients need, in addition, a specific polygenic electrical vulnerability? And the question that kills is, what is actually which type of cardiomyocyte is the true cellular substrate? Well, we hope to answer at least a few of these questions in our upcoming trial, in which most of you, and I hope all of you, will participate. I reached already out to many of you. In the TRACK-VF trial, we started recruiting. It has two parts, the clinical part and a cellular part. So we will reach out for a larger multi-center cohort of SCVF, IVF, and controls. And then this time we will take plasma samples in SCVF and IVF for different time points over a period over a year to correlate the presence or absence of the antibodies and to correlate this with outcomes. We will test the DPP6 risk haplotypes in other patients than Dutch. It has never been tested outside of the Netherlands. So control individuals will only have a single plasma sample. And we have a cellular arm where we try to create, in collaboration from blood lymphocytes from SCVF patients, to reprogram these cells into a human iPSC, and then to differentiate it into ventricular cardiomyocytes and Purkinje-like cells. So each patient gets two different cell lines, and then we will do a whole cell patch clamp and snRNA-seq to see if this will answer a few questions. So this is a little bit in a fast summary what we will do. Thanks for your attention and your time. I hope I'm not too much over.
That was wonderful. Thank you very much. So I will just ask a couple of very quick questions. This is a really exciting paper. Christian, if I understand correctly, 50% of the patients with short-coupled VF had a positive TRK antibody result.
Yes.
And if I remember correctly, about almost 80% of them had recurrent VF events, compared with the very low rates of recurrence in the other groups.
Yes.
Did you get any sense, looking at the when the time point of the sample collection was made in relation to when the VF event happened?
Yeah.
Was there any kind of signal or clues to proximity there?
So this is a very important question, because one of the questions is, when do you collect the sample? So we were lucky in my center when it's our patients. So our team already knows when there's any IVF patient and they're getting stabilized, and then their initial series of clinical tests, and then our team jumps on them. So we had a very short interval, and there was no difference between IVF and SCVF. So the average duration was 2 months. So there were a few patients that we just saw outpatient. Most of the time we already tried to collect the blood and the plasma during the index hospitalization. So we were very close to the index event. So this is one of the questions: could this contaminate? There are other papers in other cohorts that demonstrated autoantibodies, and it still showed for different electrical diseases a signature, even with a medium delay of 4 years between sample collection and the index event. So we don't know yet. So as this was very costly, I could, for the pilot study, only do a single time point. But we will do different time points, because we cannot exclude that some of these patients already cleared their plasma or had just too low antibody levels.
Okay. An additional question I have. So the antibody test was essentially a binary test result. It was either positive or negative.
Yeah.
But obviously there was 10% of the IVF population and 13% of the controls had a positive result. Do you have any idea whether or not there is a difference in titer levels between the control, for example, that were positive,
Yeah.
and the short-coupled VF
Right.
patients?
Indeed, so this is a semi-quantitative test, and we will see if we can set up an ELISA. It's very difficult because there's no commercial template for this, has never been done. So first of all, in SCVF patients, the antibody signature was always stronger than in controls. The controls, to be honest, I'm a little bit scared for these donors, which we don't know who are them, because They were healthy according to a clinical questionnaire and standard test. They were not from our centers, but we cannot exclude that these are potential candidates. We actually tried to contact the center. In the IVF patients, we had it in 10%. The question is, are these just like masked forms of SCVF? Because, everybody is IVF until we can document the VF onset, right? So are these potentially SCVF cases? The controls are a little bit troubling, but the response was weaker. And again, an SCVF patient as a blood donor, or let's say a cell donor or tissue donor, would appear healthy until he has his arrest, right? ECG is normal, echo is normal, all cardiac imaging is normal. Most of the time they have no prior syncope or anything. So it is a bit troubling. Who are these? This is why we need larger cohorts to see if this is just a little bit of background noise. So we had like we had a 2-to-1. So we had 38 controls and 38 14 plus 19 probants. So it's still a small group. So we have to see how it turns out. And again, you're right. We have to work on titers.
Amazing. Thank you, Christian. That's fantastic. We are a bit over time, so apologies for that. But again, a huge thank you to our speakers, a huge thank you to everyone here for attending. Just a few quick messages, notes for your diaries. So the next HiRO rounds will be the research rounds on March the 2nd. That's 5 p.m. Pacific time or 8 p.m. Eastern. And then big one for your diary is the HiRO Symposium. So this is the big 10-year anniversary. So save the date, please, May the 28th to the 30th in Vancouver. Thank you again, everyone.
Thank you very much.
Good night.
Good night.